So I'm going to record this session. I hope everything is clear. You can see the PPT, everything. So the subject is Immunohistochemistry and molecular pathology, and the subject code is 21 MSH 716. So today we will finalize this topic and we can go to new topic. So if you see your syllabus, in the second unit of your syllabus, you have all your immunohistochemistry. Okay, including fluorescein in situ, fluorescein isothiocyanate techniques, or immunostaining, unmasking of antigens, amplification of binding sites, or retrieval of antigen sites, then labeling of antibodies, preparation of conjugates, various types of conjugates, then blocking of endogenous enzymes, preparation of frozen sections and preparation of paraffin sections and controls. Okay, in this third chapter, they also given about importance of controls in immunohistochemistry. Okay, so apart from this, I will also keep a small topic which is called troubleshoots in immunohistochemistry. Okay, troubleshoots means if we encounter any problems during the testing procedure, how to resolve that problem. Okay, what if there is overstaining of tissue? What if there is our antibodies are unable to bind to the antigen sites? The epitopes are not so visible. The epitopes are not so efficient in order to bind with the paratope of the antibodies. So those issues will be addressed today. Okay, how to resolve the issues that we are encountering in the immunohistochemistry, that I will see. So a quick revision of all lectures. So we know what is the definition of immunohistochemistry. Basically, in immunohistochemistry, we will target an antigen with the help of primary and secondary antibodies. Secondary antibodies are conjugated with an enzyme that develop color upon reacting with a substrate to give a color. Okay, that color will give positive or negative regarding the presence or absence of that target antigen within the tissue. So this is the idea of immunohistochemistry. Okay, so there are many uses of immunohistochemistry. So right, like in cancer therapies, in infections, so there are many applications of immunohistochemistry. Then we also discussed about the basic steps in immunohistochemistry. So the basic steps are including tissue preparation, pre-treatment and staining. Tissue preparation include fixation, pre-treatment include antigen retrieval, blocking of endogenous enzymes and blocking of non-specific sites. Then in the staining, we have various types of staining protocols, right? We have enzymatic staining. Okay, we have many staining protocols that we will see. So yeah. So we have seen all the importance of these antigens, their factors affecting various types of antigens, their classifications, their usage, their preparations, their concentrations, their target organs. Then we emphasized about the importance of vapor fixation, the role of vapor fixation and the role of freeze drying or freeze fixation in the histochemistry. Then we discussed about their advantages, disadvantages, everything we discussed. Target tissue and the type of fixator that we need to use. Then we discussed about antigen retrieval methods. So we have various methods of getting back the buried antigens upon fixation. So fixation caused cross-link formations. Now we are going to remove these cross-links, and that particular step is called antigen retrieval. So we have many types of antigen retrieval methods, like we have enzymatic methods and heat methods. So enzymatic methods, here we will use various types of enzymes like trypsin, chymotrypsin, proteases. All these proteolytic enzymes will be used with the help of enzymes to retrieve the antigens. Whereas in the heat, we have many types of heating methods, like we have microwaving method, boiling method, water bathing methods. Then with the help of radio waves, we can do all these things. Okay, so all these heat-induced epitope retrieval methods we have discussed. Then and also one important point in antigen retrieval, upon heating it, we need to refold the proteins. In order to facilitate the refolding, we need to cool down the tissue. At the same time, we need to introduce an appropriate pH to the tissue. Only in that particular pH, the proteins will refold back into their original shapes. So both pH, subjecting it to the pH and cooling down the tissue is important. And also, when we are subjecting it to the pH, we will use a common buffer called tris buffer solution. So tris buffer solution is an ideal solution that have the multiple range of pHs. Okay, so in the presence of this tris buffer solution, the proteins will unfold or refold to their original state. So that is the idea of this antigen retrievals. Okay, so then we discussed about various types of okay, we also discussed about in endogenous enzyme blocking. I think I haven't discussed how to block specific enzymes. That we will discuss in tomorrow's classes. Okay. Anyway, then we discussed about this one. What is it? Blocking of non-specific antigen sites within the tissue. So that blocking of non-specific sites can be achieved with the help of using secondary antibodies taken from humans or from human serums or from secondary species, or the antibodies that we have taken from the secondary. Okay, the secondary antibody species will be used for blocking non-specific sites within the tissue. So this is the idea of non-specific siting blocking. Then we discussed about other things. We discussed about various types of staining protocols. Okay, so staining methods, we have various types of staining methods, like yeah, 1 second, I will go back. There is a picture. So here we can use various types of stainings. Either we can use fluorescent components to the secondary antibody, or we can use radioisotopes, or we can use enzymes like various types of horseradish peroxidase, alkaline phosphatases, avidin biotin complexes. So all these enzymes can be used, or even fluorescent fluorophores can be used, or even radio isotopes can be used. Okay, so yeah. So I'm going next. So depending upon the enzymatic staining, so we can conjugate various enzymes to the primary antibody or secondary antibody, such as we can conjugate with peroxidase and we can develop the color with the help of anti-peroxidases. Okay. Or we can also, and we will use various types of coloring substrates, such as enzyme will react with a substrate. So the substrates are called chromosomes. The usual chromosome that we are taking in the part of immunohistochemistry is a diaminobenzoamine tetrahydrochloride reagent, which is a DAB reagent. Okay, DAB reagent will give this brown color to the tissues. Okay, so this is all about various enzymatic staining protocols: peroxidase, anti-peroxidase, phosphatase, anti-alkaline phosphatase, anti-alkaline phosphatase methods. Then avidin biotin complex methods. Then we have polymerase chain, dextrin, polyester dextrin chain methods. So we have various methods of detection of these enzymes. Okay, avidin biotin, all these are sensitizing methods. So they will increase the sensitivity of the, or they will amplify the coloring of the tissue. Okay, so these are coloring amplifying methods. This alkaline, alkaline, and avidin biotin, all these are. Okay, so this is all about immunohistochemistry. Now, the second part of this lecture is very important. Here, I'm going to introduce 3 important topics: the importance of controls in immunohistochemistry. Then, second one is something, this one, limitations of immunohistochemistry. Okay, limitations and controls of immunohistochemistry. Then third one will be the this one, troubleshoots of immunohistochemistry. And another important thing, see, upon staining with our antibody, so this antibody localized the particular antigen within the tissue. Say for example, so in this tissue I need to target this particular antigen. So this particular antigen has been targeted with my antibody, and my antibody given color to this particular part of the tissue. Okay, now I can see only the antigen part of tissue, but I cannot see the complete morphology of tissue. The tissue is still transparent. Okay, now after immunostaining, now we need to counter stain the tissue. We need to counter stain the tissue with H&D, hematoxylin and eosin. With the after counter staining, now the tissue will be visualized. Now within the tissue, we can able to see the location of our target antigen. Okay, and that is called counter staining. Upon immunostaining, upon performing immunohistochemistry stainings, okay, now to the same tissue we must do counter staining. Counter staining must be done with the help of hematoxylin and eosin. Counter staining will give the background, the entire tissue will be visualized. In that entire tissue, now we can see the special color. Our immunostained color can be seen. Okay, so counter staining with the H&D is very important. Okay, yeah. Now coming to the controls. So what is the significance of these controls? Okay, so control means Okay, I performed IHC immunohistochemistry staining. Now I got some. Okay, my target antigen is prostate antigen, prostate specific antigen. Okay, and I got the color. How can I trust my results? How to trust that my antibody really showing me the prostate specific antigen? How to trust my antibody binding property to the tissue? For that purpose, we don't know. Maybe that might be another protein resembling like a prostate antigen, and it might bind it. We don't know to what antigen it exactly binded. We know that it is a specific antibody that will bind to specific antigen, but still we can't trust this antibody. How to prove that this is a proper binded site? How to prove this my test is correct? In order to prove the test, now what we will do is we will take known prostate antigen sample. When I say known sample, I already know that there is this particular prostate antigen. Now I will bind, I will add the same sera. I will add the same antibodies that I used in the test sample. I will use the same antibodies in my control. Okay. In my control, if I'm getting color, that indicates that this both tests are correct. Since my control got positive, that indicates that my staining is correct. If my control didn't get positive result, it is a positive control, but I got a negative result. That indicates whatever I'm seeing in the test is wrong. So control plays a very important thing. So here we will take both positive control and negative control. Positive control, here we will take actual antigen and we will go with the staining, and we will take negative control. In negative control, there won't be the specific antigen. So we will cross verify our immunohistostained section with positive and negative controls. If in positive control, we must get positive. In negative, we must get negative. If these two getting right results, means that indicates our test is correct. Okay. For this purpose, we need to use the same reagents that we used in the test. Okay. Same reagent in the test need to be used on controls. Then only we can validate the test. Okay. So that is the significance of usage of controls in the immunohistochemistry. So this is all about the controls in immunohistochemistry. Then counter staining, as I stated. So we have done immunohistostaining, then we have cross verified with the controls. Then now we need to counter stain the background. Now we need to counter stain the tissue. The overall tissue will be counter stained with HND. Okay, hematoxylin and eosin will be adopted for counter staining. So not only hematoxylin and eosin, so there are many counter stains we can opt in order to visualize the tissues. Okay, so like we have HOCHAST stains, we have DAPI stains, diamino 2-phenylidyl. Okay, so DAPI stains. So we have many types of this counter stains are there, but usually we will employ this HND staining only. HND staining is the most commonly used counter stain in immunohistochemistry. Okay, so this is all about immunohistochemistry. Then let us see some limitations of immunohistochemistry and troubleshoots in immunohistochemistry. By this, we can finish this topic. Okay, so what are the limitations of immunohistochemistry? Now, the limitations means we can say some disadvantages. Okay, disadvantages of immunohistochemistry. So there are some limitations like if we are not doing the antigen retrieval step properly in the heating procedure, if we overheated or if we less heated, there is a chance of destroying the epitopes of the specific antigen. If my specific antigens are destroyed in the process of heating, in the process of antigen retrieval, then my test will be useless. There is a chance of antigen destruction in the immunohistochemistry. So very small antigens, the peptides with very small molecular weight cannot be stained in immunohistochemistry because small peptides can be easily destroyed by heating. So that is one limitation. And you need at least 3 to 4 years of experience in handling immunohistochemistry tissues, because the tissues are really sensitive. Okay, if you do any minor mistake in this procedure, you won't get any results. So experience is a critical in performing immunohistochemistry. A fresher, a intern, a newly joined employee cannot do immunohistochemistry. There will be many errors to the fresher because he don't know the level of sensitivity that he is dealing with the tissue. Okay, simply we will think we are adding reagents. No, even the reagent concentration, the way you are adding to it, your pressures, your temperatures, the tissue status, the concentration of reagents, everything need to be considered in immunohistochemistry. Very, very sensitive methods. So experience is important. Then there is a possible antigen loss in immunohistochemistry. So there is a high chance of antigen lossage. Okay, either enzymes can destroy the antigens, either heating processes can destroy the antigens. So there is a possibility of antigen lossage. Okay, and third one is availability of antibodies. Yeah, see, now I'm telling that I have discovered a new protein and I want that protein specific antibodies. But those antibodies need to be prepared by the industry, right? If industry don't have the antibodies that I want, because my protein is very unique protein, this protein is not present anywhere. So availability of antibodies is a really challenging thing. I know that I want to detect this particular antigen, but to that corresponding antigen, no one has made the antibodies. There is no commercially available antibodies to my specific antigen, and non-availability of antibodies is also one problem. So these are the three challenges that we are facing in immunohistochemistry. And also the test, the cost of test is really high, because we already know how we are preparing antibodies, right? We are doing that by hybridoma technology. We are keeping in a rat. From rat we are taking antibodies. They are fusing, those plasma cells are fusing with cancer cells. It's a very big deal. So it is costly. We need experience. There is a possibility of antigen loss, and non-availability of antibodies are few limitations of immunohistochemistry. So this is all about the limitations of immunohistochemistry. And last and final is troubleshoots in immunohistochemistry. Okay, now I will give you some problem, and now we will try to resolve the issue. We got a problem, and how we are rectifying the problem. So that we will see in now. Okay, troubleshoots in immunohistochemistry. So coming to the troubleshoots in immunohistochemistry. So what is the meaning of troubleshooting? So troubleshooting is a systematic approach to prevent a to a problem, to solve the problem. Can be stated as a troubleshooting. Okay, so it's a systematic process of solving any problems, can be stated as a troubleshooting. So these problems can occur in the machines, in the electronics, in the computers, in the clinical testing processes, or even in the softwares. We may get the problems. Identifying the problem and resolving the problem can be stated as a troubleshooting. And let us see what are the common problems we will encounter in immunohistochemistry. Yeah, so the common problems can be three issues we will see in immunohistochemistry. One is weak staining or not at all staining. So we have done all this 1 hour procedure. So after 1 hour procedure, when we are seeing under microscope, there is no staining, or there is very less staining in the tissue. What is the reason for less staining? Then second problem, we will get overstaining in the tissue. Okay, the tissue is too much colored. What is the reason for overstaining? And what is the reason for high background visibility? Okay, so first one, weak staining or no staining. We already know weak staining is happen maybe due to our secondary antibodies conjugated with enzymes, right? So these secondary antibodies are unable to bind to the antigen. That might be the reason for no staining or weak staining. My antibodies didn't bind it. That might be one reason, right? Because of non-binding of antibodies, there is very less stain outside. That means this indicates there is a problem with the tissue antigens. My antigen retrieval step, I haven't done it properly. Because of improper or weak or okay, because of improper antigen retrieval procedure, now I'm getting weak staining. So I should go back and I should do antigen retrieval step again. That might be one reason. Then overstaining. Overstaining I may get due to our disability in blocking endogenous enzymes. Maybe tissue endogenous enzymes has been not properly blocked. Maybe those enzymes are cross-reacting with our staining enzymes, and they are overstained. Endogenous enzymes given color, and that might be the reason for overstaining. Okay, so if that is the reason, then what should I do? I need to subject this tissue again to enzyme blocking reagent. I need to incubate this in enzyme blockers before I'm going for staining. So like this, we have certain problems, and let us see possible remedial actions that we can take in order to compensate the problem that we encountered. Okay, so let us see the troubleshooting things. Okay, so one source, inadequate deparaffinization. Okay, so if there is inadequate deparaffinization, how to remove this paraffin section? So deparaffinization sections need to be subject to longer period to the xylene. We know that xylene is a very good it will remove the paraffin. Okay, so very clear, good clearing agent. So we will subject tissue to xylene solution in case of okay. By the way, I'm sorry students, weak or no staining. So that the thing is, we got weak staining or there is no staining at all. So one reason might be paraffin wax is blocking. So we need to deparaffinize the section. Then second one, inactive primary antibodies. So my primary antibodies may not bind it to the tissue. Maybe my primary antibodies are expired. My reagent may be expired. So I need to replace the primary reagents. Primary antibodies need to be replaced. Then a third reason might be antibodies do not work due to improper storage. Yeah. So you may kept the antibody reagents in improper temperatures. Antibodies are really sensitive. If you subject antibodies to high temperatures, they will easily get destroyed. Okay, that is always freezing antibodies is very important. So antibodies may be improperly stored antibodies, and other one is antibody concentration is too low. So the level of antibodies we are taking is too low. That's why they didn't bind it. Then inadequate incubation time. You haven't waited for half an hour. Rather than half an hour, after 10 minutes, you went for next step. That might be one reason. And inadequate or improper staining. Improper staining might be one reason for this weak staining. Okay, so these are the sources of error, and these are the possible solutions to the errors. Okay, this is for weak staining and no staining procedures. Then let us go to next one. Then next one is overstaining. What are the reasons for overstaining of tissue? So the overstaining of tissue might happen because of concentration of antibodies were too high. So you have used more antibodies in the reagent in the staining procedure. So that's why more antibodies binded, more staining given. So antibody concentration is high. Then incubation time was too long. So since I said half an hour, you said you may wait for 40 minutes, so you'll get good results. That results in excessive staining. Okay, so that is prolonged incubation time can cause overstaining. Then incubation temperature is too high. If incubation temperature is too high, high temperature, more filtration of dye into the tissue. Okay, so high temperatures can cause overstaining, and even wrong substrate. So substrate incubation time. So the substrate that you added to develop color, that incubation time might be overly. It is more. That is the reason why we got excessive staining. Okay, and sections dried out. So your sections has become dehydrated, and that might be one reason for overstaining. So these are the reasons for overstaining. And high background. So high background color is occurred due to endogenous enzymal activity. Okay, so those endogenous enzymes need to be blocked. So this is all about your immunohistochemistry and their troubleshoots. Okay, So yeah, these are the applications of immunohistochemistry: classification of neoplasms, malignancies, all protein related antigen targeting can be done with immunohistochemistry. Okay, so by this we have successfully finished immunohistochemistry. So let me share the notes again, and let us have some Q&A session in immunohistochemistry. 1 second, let me. Then I will go to the next topic. Let me share my PPT with you. Okay. All right. Yeah. So in a summary, if you observe clearly, students, I have kept a complete summary of immunohistochemistry in the first slide. Okay. So here in immunohistochemistry, we took a biopsy. Okay. Then this has been subjected to heat. So then we, after antigen retrieval, endogenous blockage, then we have stained with primary, secondary antibodies. Upon staining with them, we got visuals like this in the microscope. And also we have counter stained with H&E staining. So this is immunohistochemistry is all about detection of target antigen in the tissue with the help of specific antibody is immunohistochemistry. Now, there is another branch. There is another area that we need to consider in histopathology is functionality of organs. I need to check the functional status of an organ. Say, for example, I will take the heart or I will take kidney. In the heart must always pump, right? If there is any heart related problems, then heart won't, okay. If there are any heart related problems, then heart, the activity that responsible for normal heart function, those enzymes might be depleted. Listen, all organs are functioning because of enzymal activities. Every organ has its own unique population of enzymes, its own unique set of enzymes present in each and every organ. Like in my heart, I may have more SGPTs, okay, sodium, ALP and ALTs, okay. Say for example, I have more alkaline phosphatases in the heart, and I have more transaminase phosphatases in kidney, okay. So if we see the number of enzymes in the heart, ALTs will be more in heart. So one specific group of enzymes will be more in heart. Okay, now decrease of this particular enzymes, rather than having ALT 10 enzymes, if they have only 2 enzymes, that indicates because of this low enzymes, the heart has issues. That is one thing. Then second one, heart has 10 enzymes, but those 10 enzymes are not activating. Yeah. Those enzymes can be detected by our immunohistochemistry. Immunohistochemistry will detect number of enzymes in a heart tissue. Okay, that is fine. Immunohistochemistry given me a value which is normal value, which is there is supposed to be 10 enzymes in heart. I did immunohistochemistry staining, then I counted and I got 10 enzymes. Okay, but immunohistochemistry never give me the data of functional status of that enzyme. Are these 10 enzymes working in the heart or not? Maybe they are not working. Maybe they are in normal number. They are in 10. 10 is normal. But those 10 enzymes are not working. And how can I able to measure the rate of enzymal activity in that particular tissue? How to measure the functional status of the enzyme in that particular heart tissue? That question immunohistochemistry cannot answer. This is the major limitation of immunohistochemistry. Immunohistochemistry can only tell about how many proteins present in the tissue, but it won't tell the functional status of those proteins. When I say protein, here enzymes. Immunohistochemistry cannot tell us whether these enzymes are functioning or not. And it has a tremendous applications. By understanding the functional status of the enzyme, I can able to draw the conclusion of cardiac activity, the possibility of getting heart attacks in the patient. So understanding the functional status of enzymes in the tissues it has its own broad of applications and uses. And what type of methods, what type of procedures, protocols, tools I can use in order to assess the functional status of enzymes within the tissue. And that question will be addressed by another branch of histopathology called enzyme histopathology. Enzyme histopathology or enzyme histochemistry will try to give answers of this problem. Enzyme histochemistry will try to study the enzymes in the tissues. Enzyme histochemistry will try to study the functional status of the enzymes within various tissues of our body. Especially, we will target some important enzymes, such as we will target phosphatases, dehydrogenases, oxidases, esterases, and thyrosinases. We have identified that these 5 enzymes have a very crucial vital activities within broad range of tissues in the body. Most of the organs requires these enzymes for their normal activity. So understanding or detecting the functional status of these following enzymes will give us an idea about the functional status of the organ also. Okay, so from tomorrow onwards, we will try to understand what is this enzyme histochemistry, what are the types of enzymes we have, how to detect these enzymes, what type of substrates we can use. Those things we will try to study in enzyme histochemistry. Now, one interesting thing about enzyme histochemistry, here, whatever enzyme we are going to study, whatever functional enzyme we will study, we will purposefully stop that function activity. Okay, let me tell you what I'm trying to say. See, I want to see how much glucose a cell is utilizing per 1 minute. Per 1 minute, a cell must use 1 gram of glucose. Okay. How 1 gram of glucose used by a cell? It used 1 gram of glucose with the help of enzymes, glycolysis. In glycolysis, the glucose will be converted to 1,6-hexokinase will convert to 1,6. What is it? I forgot all these names in glycolysis. So the products will come, right? So there will be this hexokinase, aldolases, these transaminases, all these enzymes will activate, right? Now my idea is, now I will block this enzyme. If I block this enzymal activity, so this enzyme won't get involved in the pathway. So I will get the right answer. So if this enzyme is keep on working in glycolysis, then I cannot assess the functional activity of enzyme. So that is why what I will do is, first I will give a blocker, enzyme blocker to the tissue. So this blocker will temporarily it will stop the functioning of that enzyme. So that enzyme won't work in the pathway. So it won't go here and there in the cell. It won't go to mitochondria. It won't go to ribosomes. It will stay in the cytoplasm only. Upon blocking the enzyme, now I will go with the detection of this enzyme. So in enzyme histochemistry, definitely we will use some blockers to block the enzymal activity. After blocking the enzyme, now we will see whether this enzyme is reacting with the substrate or not. After blocking the enzyme, we will check whether that enzyme working or not. Okay. So that is how we will study the enzymes within the tissues. Whatever enzyme we want to study, first that enzyme will be blocked. Upon blocking, we will try to detect the enzyme with various types of substrates. Okay, we have specific substrates for detection of oxidases, specific substrates of detection of dehydrogenases, phosphatases, esterases, thyrosinases. So that specific enzymes can be detected. Okay. So this is how specific blockers will be used in in enzyme histochemistry. Okay, so this enzyme histochemistry we can have a deep overview on this coming week. This whole week we will focus on enzyme histochemistry. So after enzyme histochemistry, very basics are there, students, in the syllabus, which is in the unit 1, chapter 1 is all about central nervous system. So there are many cells in the central nervous system. So usually those neuronal enzymal activities and their neuronal proteins can be detected with the help of either enzyme histochemistry or with the help of immunohistochemistry. So that is why first they kept the target tissues in your syllabus. How to detect various types of neurons, CNS components. For the detection of CNS components, we have either enzyme histochemistry or our immunohistochemistry. That is how the syllabus has been framed. But I was trying to highlight this with immunohistochemistry first. So first, you know what is immunohistochemistry. Then this week we will discuss about enzyme histochemistry. Then next we will go with CNS. Then we will go to unit 3. Unit 3 is very simple. Molecular pathology means everything is all about PCR and modified immunohistochemistry only. Okay. So I hope the classes are clear. Any more doubts, students? Do you understood what is enzyme histochemistry? What is enzyme histochemistry? I hope you got some idea, right? In enzyme histochemistry, we are going to detect the functional status of enzymes. Okay. So yeah, any doubts, any confusions in immunohistochemistry? No, right? We can proceed with first unit now, which is enzyme histochemistry. So from tomorrow onwards, we can start enzyme histochemistry. Thank you very much, students. Let me end this session.